Okay, so we are going to start part 2 of our Science 10 Genetics unit. So this is the second part to section 1.1, and our first part focused on the structure of DNA, where now we are going to be moving on to the function of DNA. DNA has many functions. To start off here, DNA contains the instructions for all cell functions. And therefore, DNA indirectly controls all the functioning of all living things. So A is most likely the main function of DNA. And this is one really important molecule if we think about storing the instructions for all cell functions. B, DNA determines the hereditary traits of an individual. So the information that gets passed from one generation to the next, and so on. DNA evolves, it changes through mutations and recombinations. DNA, essentially, it changes. It looks different because of mutations or because of recombination events, where it's basically rearranged into something different. So this allows for new characteristics and abilities to appear, which may help an individual to survive and reproduce. And then finally, self-replication. So DNA has the ability to make copies of itself. And this is pretty unique because you can't think of many things that are able to copy itself. The arrangement of bases in DNA directs all cell activity. The bases are like letters that carry a message. We think of this as a code. So the bases are, again, A, T, C, and G. And so these letters in some combination create a code, and then that code gives instructions for a specific task. Normally, when we think of DNA, we think of it in the structure of it being a twisted ladder. However, DNA is stored in a really condensed form in order to fit into our cell nucleus. So DNA is stored in the form of chromatin. This is essentially the DNA is wound up and twisted. Think of like a roll of string when you untie it and then re-wrap it back up, or like the string of your sweatshirt being wound around your finger until it's really tight and compressed and it's a lot shorter than its original length. So this is essentially what's happening to DNA in the form of chromatin. Chromatin is made up of DNA and proteins called histones. So the DNA is wrapped up around these histone proteins. When a cell is growing, part of the chromatin will unwind so that the targeted section of DNA can be read to make messages that control the rest of the cell. So little sections of DNA that are important unwind from this histone protein so that the code can be accessed. But the rest of the DNA that isn't needed at that time remains wound up, and this is to avoid taking up unnecessary space within the cell. And then when cells reproduce, the entire chromatin is coiled up, and this forms an X-shaped structure called a chromosome. So this is what you were familiar with from Science 9, where the chromatin, which again contains those histone proteins, folds on itself really, really tight to form that signature X shape. Some terms that you should be aware of. So let's start at kind of the smallest, most condensed structure, and then we're going to kind of unwind this further. So in the bottom right corner, we have a cell, and the most important feature of this cell is the nucleus. So this is where all of our DNA is contained. It's the center of the cell. Then within the nucleus, especially during cell division, things are in the form of chromosomes, which are those X-shaped structures that we are familiar with. And then if we unwind that chromosome a little bit further, you end up with chromatin. And then again, if we unwind that much further and remove the histone proteins, you're left with just DNA. DNA winds up, if we're going kind of from the left-hand image all the way back to the nucleus, DNA winds up around histones, which are proteins. This forms a structure called chromatin. And then the chromatin winds further and folds upon itself, forming the X-shaped chromosomes. And then those chromosomes are found within the nucleus. Every organism has a characteristic number of chromosomes, again, those X-shaped structures. This can be observed in the human karyotype shown on the right. So the top right image is an example of what's called a karyotype, and this is simply an image of every single chromosome, which has been sorted by size. We can see it kind of going from large to kind of smaller sizes as we go through. There are complementary pairs of chromosomes. So chromosomes are always in pairs in the nucleus. This can be observed in the human karyotype shown in the right. So again, that's that top image. If we're looking up to chromosome number 22, you'll notice that they almost look identical. They're not exactly identical, but they look really similar. The size, everything about them is very similar within those pairs. These are what are called complementary pairs of chromosomes. So these 22 pairs are identical. If we look at the 23rd pair, that's this one here, they look a little bit different. And these are called our sex chromosomes. So a biologically male is XY, so that's the 23rd pair, and a biological female, based on genetics, has the chromosome pair 23 as XX. You can see that the chromosome X, if we look at that 23rd chromosome, is a lot bigger than the Y chromosome. Show you here. So this first one is the X chromosome, and then the second little one next to it is the Y chromosome. So humans again have 46 total chromosomes and 23 pairs. Different organisms have a different number of chromosomes. So the example provided here is that cows have 60 chromosomes and corn has 20 chromosomes. Another example is something like the fruit fly, which has 4 pairs of chromosomes for a total of 8 total chromosomes. So point 5 here is that genes are found on chromosomes. A gene is a small segment of DNA found at specific places on a chromosome that code for a protein. Again, as a reminder, when we're talking about protein here, we're not talking about the food you ate last night. This is strictly from a genetic standpoint that a gene codes for a protein which serves a function within the cell. Genes vary in length from hundreds of base pairs to thousands of bases. A reminder again that bases are A, C, T, and G. The arrangement of these bases determines the protein. So the order of those letters, or the order that those letters are in, and the number of letters present is going to determine what you make. So if you change a letter, say through mutation, anything like that, you change the protein a lot of times. Genes are located on the chromosome and they contain information to produce a protein. That's the big takeaway from this. And then the example here is you have 2 separate codes, this one here versus this one here. So if you look, the coding of them is different. Therefore, they are making 2 completely different proteins. So even though they're the same length, the combination of letters is completely different. So 2 different strands make 2 different things. So you can kind of think of this like we have 26 letters in our alphabet. How they're arranged determines the words, but those letters can make up different words. So that's kind of an analogy for this. Here we have a very simplified image of a chromosome showing genes. Each chromosome carries approximately thousands of genes, so tons of genes. Your body uses 90,000 to 100,000 different proteins. So that's a ton of different proteins and a ton of different things that need to be coded for. We have 22 pairs that are homologous and one pair that is different, and that's the 23rd pair. The two chromosomes shown in the image are homologous because they carry the same information on them. You see the same type of gene on both of the homologous chromosomes. So let's kind of talk through this here. So a pair of chromosomes that have the same sequence of genes is called homologous chromosomes, just as a reminder. So how many homologous chromosomes do humans have? We have 22. And the 23rd one is not homologous, they're different. Let's kind of go through each of the genes here. So our first gene is a gene for making earwax. So this is shown in red. And then we have a gene for eye color below. We can see here that the gene for earwax is shown by the same color. This means that it's an identical gene on both of the homologous chromosomes. Where if we look for the gene for eye color, one is symbolized by blue, the other is symbolized by brown, showing that while they both code for eye color, there is a variation between those two. It is important to know that this is a very simplified image. Eye color actually takes multiple genes to code, but the main thing to notice here is that these are the same gene, but they're not coding for the exact same protein. If we then move on to the green gene here, this is a gene for making a protein that dissolves the webbing between your toes before you are born. In uterus, a fetus has webbing between their toes, so think of like duck feet, and this gets dissolved before the baby is born based on a protein made from this gene. So the main takeaway here from this particular slide is that both chromosomes have the same genes present, but may have different forms of the gene. So for example, eye color here is different, it's a different form. So homologous doesn't mean identical, but it does mean that we have the same sequence of genes. Okay, so on this last slide here, we have a couple of review questions for you to work through. So I would like you to work through these today, and then we will go through the answers to these on Friday. So that is the end of this part of our genetics lesson.